I'm happy to be here. I'm talking on behalf of the photonics group. This is a group leader by Professor Paul French and Chris Dosby, and Professor McNeil, all expert in lasers, optics, and microscope for biomedical imaging analysis. I'm going to talk about a modality of imaging called single molecule localization microscopy. And this was born a long time ago. Let's say the idea was conceived for imaging excitons. This was in superconductors. And the idea was to detect tiny emissions of light at cryogenic temperatures. The idea has evolved quite a while. At the moment that you can use room temperature and document these quantum effects, let's say, at the electronic level. Then the technology is based on fluorescence microscopy. And what you can see here is a conventional wide-field fluorescence imaging. And what you are observing is, or the detector is observing, is like the average of all these fluorescent states. But with this approach, you can use high energy, high irradiation in the absence of oxygen. And when that happens, you completely put all the molecules into the darkest states at the electronic level. And then after that, you lower down the radiation. And what you start to see is single molecules. And this is the actual core of single molecule localization microscopy. Then you had to use a Gaussian fitting algorithm. There are several algorithms, but Gaussian fitting is the one that can identify all these molecules. And then you need to take many frames using a conventional microscope. The limitation is still bound by the laws of physics and bound to the optics. But once you get lots of these points, then you can render the image and has now a resolution that is below the diffraction limit. In this case, at the moment, the state-of-the-art microscope is doing up to 1 nanometer resolution based on fluorescence. Now, in the Black laboratory, we build these instruments. This is the device. The sample goes here. Here is an objective lens. And we use a source of illumination the light goes through this multi-mode fiber. And in this way, the sample is illuminated. Again, we use the irradiation that we use is probably 1,000 times the amount of energy that you use for conventional fluorescent imaging or even confocal. And this is all, initially we started by adapting commercial microscopes. The easySTORM is based on a size microscope. And we adapted with this illumination, that the illumination, instead of coming from a laser, now is coming from a powerful LED. And finally, the sample is, you have to embed it with chemistry. You make sure that the oxygen in the liquid, the biological sample is embedded, is completely removed. Then when you got these two conditions of higher irradiation and lack of oxygen, then you start to see this electronic effect of every single molecule. But having a single field of view is not sufficient, particularly if you are in the area of medical discovery or in the area of drug development, and you need to screen a lot of different molecules. Then usually the pharma is in the model of 396, 384, or even 1536 well format, when every well has a different biological sample and you are testing different conditions. Then we decide, that's not for me, by the way. Then we came with this, at the Black laboratory, we developed this, we call it the openFrame. It's a modular microscope, it's open source, and you can build it layer by layer. And we adapted this modality called Stochastic Optical Reconstruction microscopy. All these blinks, then this is the source of illumination, And we have a motorized stage, then in that way you can move, you can go into every single well of the 96 well plate. Let me continue. Then based on this, this is now how we illuminate the biological sample with a constant illumination. This is using the Alexa Fluor 647 dye, and you can see a cell here that we label with conventional antibodies, conventional fluorescent, immunofluorescent work with this technique. And when you have this higher irradiation, you can see every single photon, every single molecule emitting some photons. And then we usually take between 30,000 to 50,000 images separated by 30 millisecond integration. The data could be between 20 to 40 gigabytes videos. That's when we need HPC in order to identify all these tiny dots. Then you end up with the same image, but the image now has a resolution that is defined by the pixel. If the user, they say, I want to render my images a 1 nanometer pixel, you can get that. And as soon as you start to zoom in, you start to see in this case, the bundles of the cytoskeleton with a resolution of 10 to 1 nanometer, depending what you need. Now, this is the actual raw data. For every single point, we get like the X and Y coordinates. Then all computer vision algorithms are based on this model, but under this technology, we got a new way to analyze this. And here is the other challenge that we have for, and what we need HPC to, instead of using conventional computer vision algorithms, how to move into this format that you got the coordinates of every single molecule in your field of view. Then back in 2019, there are like a computer desktop application based on ImageJ Fiji that you use plugins. We spent almost a week analyzing a single image. And then we decided to accelerate this in a computer environment facility, in a high-performance computer environment. Then you get your raw data here. You get many frames, up to 40,000, 50,000 frames. Now you send this to the HPC environment. And in this way, we want to get all the coordinates of every single frame. The localization process is the one that requires several computer nodes in order to get all these table of localizations. And then all goes back into a single node, and that node basically renders the image. Then after that, you can decide to increase the amount of nanometers or the resolution per pixel, or go to more cluster analysis. And let me dive into that. Then now we can make these images, but we want how to apply this into real problems in medicine. Then it's not showing anymore here. Yeah, there it is. Then still, in kidney disease, you still need electron microscopy to stage different diseases. And one of these is the diseases of the glomerulus. It looks more like a digital cauliflower or something like that. It's full of loops, but this is the way that the blood is converted into urine, and it could be attacked by immune complexes, antibodies, so immune diseases can generate this problem. Then I'm showing here the conventional electron microscopy. This is the glomerular basement membrane, and in this disease there is usually measured around 200 nanometers, but in the disease that layer gets enlarged, and this is called minimal chain kidney disease. Then this sample is actually in a paraffin sample, in a paraffin slide, and we basically use antibodies to document the basement membrane with an antibody, and here you see the raw data. It looks better with the dark light, but yeah, you can see all the different molecules now coming from the antibody. And when you localize, this is the resolution, let's say the full width and half maximum with a 100x objective. But as soon as you get into super resolution, you get a more high result image. Then at the moment, at this stage, we went to the expert in kidney disease that uses electron microscopy. Candice Rofusa in the Hammersmith Hospital, she can use this data and could be as similar as you can use the electron microscopy. And we were very pleased about this. Then we moved to other diseases. Then here I show you membranous glomerular disease. Again, this is the whole glomerulus under 20x, and you use antibodies. In the red channel, we use antibodies to detect the immune deposits that are actually causing the disease. And this is the actual, you use a reference that is the membranous, the glomerular membrane baseline. And when you move to 100x, I mean, it's very easy when the expert tells you, look, I need to look at this area. And in this area, this is what you barely can see. But as soon as you go into super resolution, you can see in red now this deposit with greater resolution. And this image is also, we choose this area here in electron microscopy. These dark areas is equivalent to these deposits here in red. And here it is again. This arrow is the annotation of the pathologies. Then here is the immune deposits, and this is the reconstruction with fluorescence. Then this sample takes about a day to prepare manually in the lab with conventional immunofluorescent techniques, primary antibodies. But a sample for electron microscopy could take up to a couple of weeks, just the preparation and the actual imaging of the electron with electron microscopy. We also choose another disease. This is lupus nephritis. And again, these areas were chosen by the pathologists. Here is the area of immune deposits. And again, you can see in 100x, I mean, you barely can see any detail. But under super resolution, you start to highlight where are these deposits. And this was useful for the pathologists. Now, this is only one single slide, and again, in pharma, you need this loss of field of view, 96 well format. Try to bring single molecule localization microscopy into high content analysis. And in this 96 well platform, you can see here a drug screening. This is a grid of every well has, it's the same cell line, but with different drugs, challenged with different drugs. In this case, siRNA interference. And here is the wide field image, and here is the super result image. Here is trying to analyze what happened with the cell move, the cell migrate, and the cell can experience traction against. Then what you see in green is the focal adhesions making traction on a surface that is a glass surface coated with fibronectin. And yes, let's say the expert, that is Professor Chris Bacala, ICR, can see some patterns, but here is where the challenge comes. When you don't have expertise or you don't have knowledge about it, how to analyze this different. Then let me show you. This is called the nuclear pore, this structure that you see here. And under 100x, you barely can see through them. In the blackhead unit, we've been changing the different detectors. Then you can have detectors, expensive CMOS camera, or other detectors from with different electronics as well, but with lower cost. And what you see here is the nuclear pore. Is in the nuclear envelope in a cell. It's actually a unit structure. There is lots of crystallography data to support that this nuclear pore has 8 subunits. It's like a window and the diameter is around 100 nanometers. Then this is around 100 nanometers. But when you are in super resolution, you start to see the subunits. And when you just plot an intensity profile around every donal, let's call it, then you can start to see that there is an improvement in resolution. Then since then we have moving into, I mean, you can image anything that you can label with fluorescence. You actually can go into this application of super resolution imaging. Then here is a preparation of chromosomes. The dye, we can make a synthetic nucleotide that is fluorescent. And here is the video of under this illumination, you get this blinking. Then this is the 100x imaging, and when you do super resolution, you see a much crisper rendering of these structures. But having the image is not enough. We know this, at the molecular level, the structure has certain patterns of organizations. Doing this by eye, it might be very tedious and difficult. Then we are now moving into ways to do cluster analysis based on points. The amount of points that you get, in this case, from a single chromosome could be quite significant. And then we think one of the gold standard cluster analysis is based on Bayesian cluster analysis. For a single chromosome, it could take a couple of hours in a 1 terabyte RAM machine with 48 cores. Then the HPC is making practical to analyze the pattern of this point. But this is in a tiny area. When you go to area like 100 microns in diameter and you want to make like clinical applications, then this type of computer, this need for computer resources is no longer practical. One of the, with Professor Magnisa Hammersmith, one of the ideas is to study the chromatin, how the chromatin is organized in cancer, and in particular ovarian cancer. This is how a cell is stained in blue or green. Here you can see the cells that are in the S phase. And in red is the histone tree. We are trying to then how to see all the different architecture of the genome architecture of these cells using single molecule localization. Then here is the wide field image, 100x, and this is a super resolved image. Then one of my colleagues in the unit has taken every single nucleus and then make a 1.6 micron square subfield region of interest, and then send this to a single computer node in the HPC environment. And it takes around 2 to 3 hours just to analyze one of these little squares here. What you see here is how the chromatin is organized. The chromatin is very dynamic, and depending when you fix the cell, you can start to query the status of that cell. And mind that we can do this in 96 well format, and in every field of view, you can get up to 6 to 10 cells. Then HPC is making practical to discover what's going on in there, but with a lot of resources and a lot of time in order to find out an answer that is, let's say, it's not practical at the moment. Then, yes, you can, in this point pattern analysis, you can apply replace K. That is probably one of the oldest algorithms to study point pattern distribution. And what you can see here, I want to take your attention to here. You see an untreated cell under replace K, but when you treat with we call it a chromatin opener, then you change the architecture of the chromatin in the nucleus. Then you start to see that there are different patterns. Now, this type of analysis is not using the image, but it's using the long table of localizations. And this is the new challenge that we have under this imaging modality for computer vision. Then at the moment, this Bayesian cluster analysis can use the points, but can you also use the uncertainties of the fitting algorithm to identify every single molecule. Now, with the help of the high-energy particle physicist, we are implementing that Bayesian cluster analysis into C++ application for HPC. All that you have seen here is in 2D. Now, moving into the field is moving into 3D, and also doing this type of analysis in 3D demands more new algorithms and new applications as well from the point of view of analysis. I think this is all what I have to talk today, and I'm happy to take any questions if you have.